All right, so today's notes are on meiosis. There's been a request for Morpheus to say hi. Hi, guys. Hi. All right, he doesn't want to stay around too much longer. All right, so today's notes are on meiosis. This is your genetics chapter 3. Section 3, these notes are a little bit longer than the previous two. All right, so first we need to review what a homologous chromosome is. This is really important throughout most of this unit. So homo means same. Logous means structure, so these are chromosomes that have the same structure, the same length, the same centromere position, and the same banding pattern. So all of our somatic cells or our body cells are diploid, which means they contain homologous chromosomes. They have 2 of each set. Out of our 46 chromosomes, we have 23 pairs, but 22 of those pairs are homologous chromosome sets. Your X and your Y chromosome are not homologous. They are not the same size and don't have the same structure. All right, so today we're going to talk about meiosis. Meiosis is a reduction division of the nucleus. The whole point of meiosis is to form haploid gamete cells. So we're trying to form our egg and our sperm cells. Somatic cells, body cells, like I just said, are diploid. They have a full set. So in humans, we have 23 pairs. Therefore, a diploid cell would have 46 chromosomes. These chromosomes are going to replicate in the S phase of interphase, and this is going to allow them to have pairs of sister chromatids joined at a centromere. Meiosis is a little bit different than mitosis in that there are 2 divisions. In meiosis 1, we're going to separate those homologous pairs. That's the reduction part. And then in meiosis 2, that works very similarly to mitosis, where we will separate the sister chromatids from each other. And what we get at the end is from a single somatic diploid cell is 4 individual haploid cells. All right, so here is kind of a visual of what's happening. We start with our one set of DNA, the one from your mom and the one from your dad. Those replicate at the beginning of meiosis 1. They split, so we get 2 cells with just 1 chromosome, 2 sister chromatids. And then in meiosis 2, they will split. Those sister chromatids will split apart, so we end up with 1 individual chromosome in each cell. All right, so we start with interphase. Interphase is, of course, getting ready for meiosis. In interphase, just like in mitosis, our cell is going to get bigger, our organelles are going to be made and duplicated. But we also have our S phase or our synthesis phase, and that's where our DNA is replicated. So each single chromosome becomes a pair of sister chromatids, and they're joined at the equator by the centromere. So here we have our 4 individual chromosomes, and then here you have them all having been replicated, so 2 sister chromatids held together by a centromere. All right, then we go into prophase 1. In prophase 1, those homologous chromosomes are going to associate with each other. They're going to kind of pair up. And we have something called crossing over that can take place between the non-sister chromatids, or the 2 chromatids that are next to each other but not attached at a centromere. This crossing over is going to result in a new combination of alleles, or a recombination is what it's typically referred to as. And that is a source of genetic variation. Other things that are happening in prophase 1 is your nuclear membrane breaks down, and the centrioles migrate to the poles, either north or south or east or west, and your spindle fibers start to develop. All right, so let's talk about this synapses, or this joining of the chromosomes. So the chromosomes are going to pair up in their homologous pairs, and this results in what's called as a bivalent. It's also known as a tetrad. So what I refer to it a little bit more often as a tetrad than a bivalent. But remember that chromosomes are the thread-like bodies that have both the chromatin and the genes, and then the sister chromatids. Each side here, this is one sister chromatid, this is another, is the identical copies of a chromosome. So when the two homologous chromosomes join together, that's through synapses when they associate with each other. So then they form their little pair, their little buddies now. And then we'll have the crossing over. This crossing is actually called a chiasma. And this is when neighboring non-sister chromatids, so the sister chromatid from the pink side and the sister chromatid from the blue side, are able to cross. This could happen at the bottom here or the top, or it could happen at both places. And this junction is called a holiday junction. And the result is when that junction is over, that the alleles are swapped between those non-sister chromatids, and that's called recombination. All right, so this is another way of writing it. This is actually the standard notation for writing genotypes of alleles on linked genes. So that is on a section of a sister chromatid where they will be inherited together. So here on the pink side, you have a big D and a little e on both of our sister chromatids. So this was your original chromosome, and then it was replicated. So we have two a big D and a little e, and then a big D and a little e, and then the lines represent the sister chromatids. The chromosome from the father has a lowercase d and an uppercase e. So that was the original chromosome. It's replicated, and now we have a little d, big E, little d, big E. So when you're looking at just this notation, the ones that can be crossed are going to be these here, and that's what we see in this this writing here. So what you can get, these will be the homologous, the new homologous chromosome combinations, and then the results for our gametes are different. Now, don't stress too much about not understanding this part. We're going to get to some activities in class where we look at this. All right, so then we go from prophase 1 to metaphase 1. The tetrads or bivalents, the homologous pairs, line up in the equator. They can be randomly oriented, so the way that they face, and this is going to lead to even more genetic variation than the crossing over did. Think about it. We have 23 sets of chromosomes. Each set has 2 chromosomes in it, so that's 2 to the 23rd possible orientations in humans. It's well over 8 million combinations. So you can see here are just some of the combinations with how they could have lined up in this particular metaphase 1. So we have 4 different ways. That's going to lead to a lot of variation in the outcome of the gametes. All right, in anaphase 1, those spindle fibers are going to contract, and this is going to cause the homologous pairs to separate and be pulled to the opposite poles. This is really the beginning of your reduction division. Something called Nondisjunction can also occur here. That's where the chromosomes fail to separate properly, and you get a not normal number of chromosomes, so your individual gametes can have more or less chromosomes. In telophase 1, the new nuclei are going to come back, and the cytoplasm is going to begin to divide by cytokinesis. The nucleoid are no longer diploid at this point. They are haploid, even though each chromosome has 2 sister chromatids. So the crossing over and the recombination has already occurred, so these sister chromatids are not going to be exact copies of each other. All right, so we do have a slight phase of interphase, but not as much as happening, and we definitely do not have any synthesis of our DNA. We just have the preparation for another division. In prophase 2, your nuclei are going to break down, your centrioles are going to move to the opposite poles, your spindle fibers are going to start to grow, but the big thing about prophase 2 is that there is no crossing over occurring. In metaphase 2, the pairs of sister chromatids are going to line up along the equator. The spindle fibers are going to lengthen and attach to each side of the centromere. We're still going to have some random orientation, but there's not going to be as much variation as we had in metaphase 1. This is because the chromatids are not having any other crossing over. All right, in anaphase 2, the spindle fibers are going to contract and the centromeres are going to be broken apart. That's going to allow the sister chromatids to be pulled to the opposite poles. Again, we could have another instance of non-disjunction where they do not break correctly, and we could have two gametes having the wrong number of chromosomes. In telophase 2, our 4 haploid nuclei are reformed or formed. Cytokinesis is going to begin splitting the cells, and what we get is 4 haploid gamete cells. Fertilization of these haploid gametes are going to produce a diploid, because 1 haploid plus 1 haploid is a diploid, and that's going to give us a cell called a zygote. All right, so this is a great chart to show the process of meiosis. Hey, great essay question. Remember, to get that 1 point, you would need to talk about the difference in interphase or any of the phases in both meiosis 1 and meiosis 2. So I'm not going to go over this, so this is a good chart to review. All right, so this image has caused much strife to children, students in the past. This image has, or something similar, has appeared on exams before, and hint, hint, you may see it in your future as well. So this is a rough image of chromosomes forming a chiasmata, or forming chiasmata, not a chiasmata. And you need to look and see how many structures you can see, how many chromosomes, how many centromeres, how many sister chromatids, and how many chiasmata. So I'll give you a second to see what you think you can find, and then I'll show you how to decipher this image. All right. I personally find that the easiest thing to start with is, where are my centromeres? Remember, your centromeres hold 2 sister chromatids together. So you have 1, 2 chromatids. So those are there. So if you have 2 centromeres, you have 2 chromosomes. Each chromosome has 2 sister chromatids. So you have 1, 2, 3, 4 sister chromatids. And the chiasmas are where there is crossing over. So you can see 2 crossing over, or chiasmata spots. All right. All right, so the whole point of genetics is to increase variation, and we have a few laws from our lovely guy Mendel, who are important in increasing our genetic variation. And the first one is Mendels Law of independent assortment. And this states that the presence of allele of one of the genes in a gamete has no influence over which allele of another gene is present. So basically, you can see here, out of this combination of parental chromosomes, we can have different combinations in our gametes at the end. So you could have a big A and a big B, or you could have a big A and a little b, or a little a and a big B, or a little a and a little b. So it doesn't matter if you have one big letter. That does not mean that you will have the allele that matches. This really only works for unlinked genes, so these are genes that are further apart on the chromosomes, or just on different chromosomes in general. All right, so also we have an idea of random orientation versus independent assortment. So random orientation, we talked about a little bit with how they behave in metaphase 1, how they line up, or how the sister chromatids are arranged in metaphase 2 in meiosis. Independent assortment is going to refer to the behavior of the alleles. Okay, so due to the random orientation of the chromosomes in metaphase 1, the alleles have become independently assorted into gametes. So they are linked but they don't have any influence over another. So you can have all these different possibilities. Okay. All right, so how do we get genetic variation? Genetic variation is really important. It's what keeps us different from our siblings, even when we come from the same parents. So we have crossing over. This is going to lead to a new combination of alleles. Our random orientation in metaphase 1 is also going to lead to more variety. Random orientation in metaphase 2 is going to give additional genetic variation. And then, of course, we have even more variation when we have random fertilization. So which little sperms, which swimmers get to the egg and are able to fertilize it. All right, so let's go back to this idea of nondisjunction. Nondisjunction is when chromosomes fail to separate properly. And I have in this image the process of meiosis, both meiosis 1 and meiosis 2, and with normal separation. All right, so nondisjunction is going to lead to changes in chromosome number. This can happen in anaphase 1 or anaphase 2 because in anaphase, either the chromosome of the chromosomes of a pair or the individual sister chromatids are pulled apart. And you can see here that instead of this yellow guy going to this pole over here, it is being incorrectly pulled to this pole. So what that means is we will have less chromosomes over here. So these two haploid cells are missing chromosomes, and these haploid cells have too many chromosomes. All right, so the previous image was a nondisjunction in anaphase 1. Now let's look at a nondisjunction in anaphase 2. So we have everything separating correctly. So we have 2 chromosomes in each cell, each with the sister chromatids. So now when they fail to separate, this blue guy is going to get pulled to the wrong side. So we're going to have one that's missing one and one that has more. But these guys over here are going to separate correctly. So these will be normal. So nondisjunction can lead to changes in the chromosome number. In the first case, all the gametes were affected. And in the second case, if it happens in meiosis 2, just half of the gametes will be affected. All right, so what does that mean? What happens if we have nondisjunction? Well, in this case, we'll have something called a trisomy. Tri means 3, so we're going to have 3 chromosomes. So nondisjunction, we have this egg here who has an extra chromosome, and it's going to combine with this sperm that has a normal number of chromosomes, okay? And then when we get together, we have a trisomy. We have one complete pair, but then we also have a trisomy. So the resulting zygote and then the somatic cells with a trisomy of one chromosome. And this can be very fatal or could cause a disorder. The most common disorder, the one that most people are familiar with, is trisomy 21, which is Down syndrome. There's a couple links here you could check out. This can happen because of nondisjunction in either anaphase 1 or anaphase 2, leads to an extra copy of chromosome number 21 in the gamete. This is usually because of a trisomy or a nondisjunction in the egg formation. And then the fertilization by the sperm is going to add the homologous chromosome number 21, which means it'll have 3 of that chromosome. Down syndrome has very distinctive physical characteristics, and patients have a slightly shortened life expectancy. They have many learning difficulties, but they are generally happy in nature. All right, maternal age in Down syndrome. So as women age, there are greater instances or cases of Down syndrome. So mother's age is a high risk factor in Down syndrome. It's because girls are born with precursors to all the eggs that they'll ever make. So beginning with puberty, these eggs start to mature, and this continues until menopause. Okay, some of those eggs, the older they are, have been there all the life. So the division may not happen. May happen with more instance. Are you coming up? All right. Frank would like to say hi. Say hi, Frank. Okay. Hi, Frank. I'm giving a movie.